right. So when Darwin presented his theory of evolution by natural selection, he talked about phylogeny, the family history of groups. In particular, he was talking about animals. So we're going to talk about animal diversity and how we make sense of it. So these are the major groups of animals, the phylum, and we're going to talk about what makes an animal, and what are the key characteristics that allow us to determine the relationships among them. And so, first, we should begin by talking about what is an animal? How do we recognize animals? What features do they share? And what makes them different from other organisms? What features do they lack? And so, again, in science, we're looking at evidence, and the evidence here is shared features. We're looking as best we can at living and extinct animals. We look at the anatomy of the animals and particularly their development, the study of their embryos, and then now genetics, the sequence data. In general, it's pretty easy to recognize animals. We all kind of intuitively know, but let's talk about it. First of all, animals are heterotrophs. They ingest food. They don't do photosynthesis. So they are they're all united by having some sort of mechanism for grabbing and digesting pre-existing food. They are multicellular eukaryotes with large, complex bodies. Even microscopic animals are multicellular. Okay, and their cells lack cell walls, which separates them from two major groups, the fungi and the plants. And so that's enough to get them down to their group. Another big characteristic is that they are active. They move quickly, and this might be their most striking feature, although some of them aren't really doing much movement. But so they have tissues for this kind of movement, and muscle and nerve tissue, which allow them to respond quickly. Okay, and so then they're diverse in form, diverse in habitat. They are pretty strict on asexual reproduction. There's some that can reproduce asexually, but the animals generally reproduced by sexual reproduction. And then, really important, there's these features of development that unite them that we're really going to talk about a lot. And so that they form a blastula that kind of rearranges. It's a hollow ball of cells to form an opening, the blastopore, which becomes either the mouth or the anus, and they form a gut during this after this formation of this pore, and that's called a gastrula. And then, as I mentioned, they have nerve and muscle tissue, unique among all organisms. So we're trying to put together a family tree of the animals, okay? And so we're going to look at these relationships. The three domain family tree I showed you, the eukaryotes are the youngest group. They're the most derived, and these include animals, green plants, fungi. Those are all multicellular eukaryotes, and then it also includes a bunch of other single-celled eukaryotes. And so that's kind of the big picture. But what does an animal phylogeny look like? It's going to show the relationships and be based on evidence. And one of these most unusual pieces of evidence is embryonic development. All multicellular animals start out as a fertilized egg that goes through mitosis, okay, which we call cleavage. Okay, and cleavage of the egg is going to produce a multicellular structure. And animals do it in two ways, radial or spiral cleavage, which I'll show you in a second. And this divides the animals into two great groups. And this radial and spiral cleavage is also the same thing that divides them into determinate and indeterminate cleavage. Then they're going to produce this hollow ball of cells, the blastula, that forms or have a rearrangement that forms an opening, the blastopore. And that too has different fates in different groups of animals. And after you have this hollow ball of cells rearranged, you form germ layers and body cavities. So let's kind of look at the diagram here. So there are two great groups, the protostomes and the deuterostomes. Okay, when the Fertilized egg divide first divides into 2 cells, then it divides into 4 cells. And by this division, we can see, okay, that it's already different in some animals than others. In the animals that have spiral cleavage, the cells divide into tiers that are just offset. So the lower layer is kind of twisted from the second layer, whereas in animals with radial cleavage, the tiers are stacked right on top of one another, and so that's deuterostomes. Okay, and then even at this early 4 cell stage, there's different genetics going on in these different groups of animals. If the In the protostomes, if you take away a cell from that 4 cell stage embryo, development fails. The they have what's called determinate development, and those cells are already different from the other cells, and so if you take one away, the embryos are defective and they die. That's not the case with deuterostomes. With deuterostomes, if you take one cell away, it's got all the potential to do what the rest of it does. And so if you split the embryo at the 4 cell stage or the 8 cell stage, what you wind up with are 2 embryos, one larger than the other. So this is called indeterminate. Okay, so then you get this hollow ball of cells, and there's a movement, a rearrangement that happens, and this movement produces an opening called the blastopore. And in the protostomes, this becomes the mouth, and in deuterostomes, this will become the anus. Okay, and so it will continue all the way through. It's the developing gut, and this movement through there will form layers of cells, which will be the germ layers. And most animals will form 3 germ layers. The outer layer of the blastocyst will become the endoderm. The inner layer of the gut will become, or that's the ectoderm, excuse me. The inner layer of the gut is the endoderm, and then a third layer, the mesoderm, forms in between them. And so this is how most animals form. The vast majority of them are going through these stages very early on. Then with animals, we also talk about symmetry, whether they have bilateral symmetry or radial symmetry. Essentially, in radial symmetry, they're a ball, and in bilateral symmetry, they develop a front, a back, a top, a bottom, a left, and a right. And so there's only one plane of symmetry that divides it into an equal half, the mid sagittal. Okay, and so then the symmetry is a big deal. I've kind of mentioned body cavity forms in the mesoderm, and there's those 3 germ layers. So we get these anatomical features that we see in animals, and generally the body cavities. There's some animals have no body cavity. They're just solid tissues: ectoderm, mesoderm, and endoderm. These are acoelomate. Coelom is the body cavity, so they don't have any. There's a pseudocoelomate, where the mesoderm is kind of attached to the ectoderm, and the endoderm floats in that. That's one group of animals. And then in true coelomates, you have a body cavity lined by mesoderm. The body cavity develops within the mesoderm, and this is the vast majority of animals that will do this. Okay, so the those are features we look for in living animals, and sometimes what we can see in fossils. And then what we do is we look at the sequence of one gene that we know all animals have, and that's the ribosome. So it's carried in all cells. We can easily get this one gene, and we know this gene probably doesn't have any horizontal transfer. And so looking at this, we wind up with um, an animal phylogeny that kind of divides the animals into these large groups of protostomes and deuterostomes, and then a few other groups that are don't have those body cavities or 3 layers or bilateral symmetry, and that's where the hard part comes in. So the true animals we talk about as eumetazoa. The other animals are parazoa, and these include the sponges, porifera, and now Cnidaria is in there. because they lack some of the things the true animals lack. So in the animal phylogeny, um, we have the eumetazoa, the true animals with the deuterostomes, all bilateral symmetry, and the protostomes, all bilateral symmetry. These groups are, the protostomes are divided into the ecdysoa. These are animals that have a cuticle for their skin and molt, like insects, crustaceans, and roundworms. Then there's the Lophotrochozoa. They have a certain larval type or a feeding structure called a lophophore. This includes annelids, mollusks, um, and lots of other invertebrate animals. And then the deuterostomes, which are the chordates, us, all the animals with backbones, and echinoderms. Okay, so this is our little taxonomy. Most taxonomists agree the animal kingdom is probably monophyletic. There are a bunch of theories for this, and saying that they may have come from single-celled protists, but we really don't know. What we do know is that um, the great diversity of animals appeared during the Cambrian period. All the major groups appeared during this time, about 540 to 525 million years ago. And all these animal groups have what are called the homeobox or Hox genes. And these are development genes that appear, and there are several units of them, and they appear and they control the development of things like symmetry, limbs, segmentation, and these are gene switches for DNA binding proteins that are turned on during development. And so you can turn these on to produce body structures like the head or the thorax or the abdomen. And you can see which genes are active here in this fly. And when these genes are active, they turn on a series of genes that result in the formation of a structure like a wing or a leg or a head. Okay, we know all the modern groups, or I shouldn't say modern groups, we know all the major groups of animals have these Hox genes, these switches. But we know the sister groups don't, like the sponges and the cnidarians, they lack them. And so these crucial genes, I'll talk more about later, and their role in the phylogeny. So we're just introducing this and introducing the developmental stuff for right now.